Okay, so welcome back to this next video in which we are discussing receptor tyrosine kinases. Okay, so we're currently in the process of looking at some of the pathways downstream of receptor tyrosine kinases. Okay, so we've seen that the activation of the receptor tyrosine kinase results in the activation of this monomeric G protein, RAS, which is attached to the underside of the phospholipid bilayer by lipid moieties. Okay, then what will happen is the RAF kinase enzyme, which was previously in the cytoplasm and inactive, will come and associate with the activated RAS monomeric G protein. Okay, this will allow then the protein phosphatase 2A to remove these phosphate groups which are attached to inhibitory phosphorylation sites. And when you remove those, the 14-free protein, which is also inhibiting the RAF kinase enzyme, will then fall off. Okay, once that's gone, what you can then do is phosphorylate activatory phosphorylation sites, and these are tyrosine residues generally, and the enzyme which phosphorylates these tyrosine residues is a tyrosine kinase, and it's one of the SARC family members. Okay, right, now what we've got is this activated RAF then. And RAF is now going to act as a serine threonine kinase, and it's going to phosphorylate the next enzyme along in this MAP kinase module. Okay, right, so before we go any further, let me just explain what a serine threonine kinase is. Okay, so we've talked about tyrosine kinases, okay, which are enzymes which are capable of adding phosphate groups onto tyrosine residues, but we haven't yet seen serine threonine kinases, and these are kinase enzymes which are capable of adding phosphate groups onto serine and threonine residues. Okay, so let me just remind you of the structure of a serine residue and a threonine residue. So once again, we'll start with the core amino acid structure, and we're showing this as residues rather than pure amino acids. Okay, so we've got an amino group here, then we've got an alpha carbon with a hydrogen coming off it, and the carboxylic acid group here. Okay, and both of these, both this amino group and this carboxylic acid group, I've drawn them as though they're attached to either a carboxylic acid group in the case of this amino group, or an amino group in the case of this carboxylic acid group. Okay, now the R group then of a serine residue consists of a methylene group, like so, with an alcohol group coming off like so. So this is a serine residue here. Okay, now threonine is slightly different. Once again, we'll draw the core amino acid structure. So here's the amino group, the alpha carbon, the carboxylic acid group, again drawn as though it's involved in a peptide link. And then the R group of threonine consists of a carbon, which then has a methyl group coming off it. Also the same alcohol group that we had off here, and then a hydrogen. Okay, so it's almost identical to serine, but for the fact that we have this additional methyl group. Okay, so those are serine and threonine residues. Now, we can add phosphate groups onto the alcohol groups of these serine and threonine residues in the same way that we can add phosphate groups onto tyrosine residues. So let me remind you of this. So let's draw our phosphate group here. So it consists of a phosphorus atom double bound to an oxygen atom, with two alcohol groups coming off either side, and then a single bond to an oxygen atom that has then acquired an electron via ionic means. Okay, and remember this group here with a phosphorus atom double bound to an oxygen atom, then with an alcohol group coming off, that is analogous to a carboxylic acid group. In fact, if that phosphorus atom was a carbon, that would be a carboxylic acid group. And it can now interact with the alcohol group on either the serine residue or the threonine residue in pretty much the same way as a carboxylic acid group would. Okay, so we're going to form a phosphoester link here. We're going to take the alcohol group off the phosphate group. We're going to take the hydrogen off the alcohol group. We're going to combine these two things together to produce water, and we're then going to bind the oxygen of the alcohol group on the serine or threonine residue to the phosphorus atom on the phosphate group. And this link that we are forming here, okay, this is known as a phosphoester link. Okay, right, so we can attach phosphate groups onto serine and threonine residues via phosphoester links. Okay, right. So the RAF kinase enzyme is a serine-threonine kinase. It can phosphorylate both serine and threonine residues within proteins. Okay, so it's now going to phosphorylate serine residues that are within the active site of the next enzyme along in this MAP kinase module. Okay, so what is the next enzyme along in this MAP kinase module? Well, I'll draw it down here. Okay, so it's an enzyme that's called MEK. Okay, so this is called MEK. Now, what does MEK stand for? MEK stands for the mitogen activated protein kinase, MAPK, okay, //ERK, which is another name for the mitogen activated protein kinase, as we'll come to, okay, and then it's kinase. So the M is for mitogen activated protein kinase, the E is for ERK, which is another name for the mitogen activated protein kinase, and then the final K is for kinase itself. So this is an enzyme which is going to phosphorylate serine residues, not serine residues, but it's going to phosphorylate residues within the MAP kinase ERK enzyme. Okay, so this is the MEK enzyme. Now there are two MEK enzymes. There is MEK1 and MEK2, and again it does not really make much difference which one we're talking about. They both do the same thing. They're both going to be activated by RAF kinase enzymes. Okay, so MEK is floating around in the cytoplasm, and what's now going to happen is our activated RAF kinase enzyme, when it comes into contact with a MEK enzyme, is going to phosphorylate serine residues, specifically serine residues, which are within the active site of the MEK enzyme. And when it gets these phosphate groups added on to serine residues within its active site, this is going to activate the MEK enzyme. Okay, so now we've got this enzyme activated, and it's now going to phosphorylate residues within the MAP kinase ERK enzyme. Okay, so before we move any further, let's just discuss what type of kinase enzyme MEK actually is. So MEK is an unusual kinase enzyme because it's actually capable of phosphorylating not only tyrosine residues, but also serine and threonine residues. So it's actually capable of adding phosphate groups onto tyrosine, serine, and threonine residues. Okay, so it's known as a tyrosine and a serine threonine kinase. Okay, now these are actually quite rare. Usually a kinase enzyme is either a tyrosine kinase or it's a serine threonine kinase. But in the case of MEK, it can phosphorylate all three. Okay, and this is going to be important with regards to its activation of the next enzyme along. Okay, so let's say we've got our next enzyme along here. Okay, and this final enzyme along in the MAP kinase module is MAP kinase itself. Okay, so the mitogen activated protein kinase, which is more commonly called ERK. Okay, now I'll tell you what ERK stands for in a moment, but for now let's just colour in MAP kinase /ERK in green here. Okay, so what does ERK stand for then? ERK stands for the extracellular signal regulated kinase. Okay, so the E is for extracellular and also signal. Okay, so the extracellular signal, and then the R is for regulated, and there is a dash in between signal and regulated. So this is the extracellular signal regulated, and then the final K is for kinase. Okay, so that's what ERK is short for. Now, again, there's more than one type of MAP kinase ERK enzyme. Okay, so there are two forms. There is ERK1, the extracellular signal regulated kinase 1, and there is also the ERK2, the extracellular signal regulated kinase 2. Okay, now again, we're not going to fuss over this because both of them pretty much do the same thing. Okay, and both of them are going to be phosphorylated by MEK enzymes of either MEK1 or MEK2 and become active. Okay, so once again, MAP kinase ERK enzymes are sitting within the cytoplasm currently inactive, and they're going to be activated by the now active MEK enzymes over here. Okay, so what's now going to happen is MEK is going to phosphorylate MAP kinase ERK enzymes on 2 places. Okay, so it's going to get phosphorylated twice. One of these residues is going to be a tyrosine residue. Okay, so this is where it's important that the MEK enzyme can phosphorylate tyrosine residues, and the other is going to be a threonine residue. Okay, and this is why it's important that it's also a serine threonine kinase as well. Okay, so here are these 2 phosphate groups we've now added on to our MAP kinase ERK enzyme, and now it's going to be active itself. And it's then just a plain old serine threonine kinase, so it's not as fancy as MEK. It can't phosphorylate serine, threonine, and tyrosine. It can only phosphorylate serine and threonine residues, which is more normal. Okay, right. So now I just would like to introduce you to a bit more terminology, because one of the most confusing things about this MAP kinase module is the fact that all of these enzymes have a ton of different names, and there is just one other naming system that I'd like to tell you about for MEK and RAF. Okay, so firstly let's start with MEK enzymes. So the MEK enzymes are also occasionally called the MAP kinase enzymes. Okay, so often this is written MAPKK, like so. So because this enzyme is the kinase which phosphorylates the MAP kinase over here, we can call this the MAP kinase, basically. So MAP is for mitogen activated protein, remember. The first K will be for kinase, and the second one will also be for kinase. Okay, now this is also sometimes denoted MAP2K. Okay, so rather than just putting 2K, some people will write 2K. Okay, so all of those names are other names for MEK enzymes, basically. The MAP kinase or the MAP2K enzymes. Now, it gets even more ridiculous when we go back to RAF, because RAF, this is now a kinase enzyme for the MAP kinase. So we can also call RAF enzymes the MAP kinase enzymes. Okay, and this is sometimes denoted as MAP3K rather than just writing out the 3 Ks. So those are other names for the RAF enzymes, and you may well see this silly nomenclature used occasionally, and don't let it confuse you. Okay, the most common names are the first ones that I gave you: the RAF kinase, activating MEK, activating MAP kinase or ERK. Okay. And to be honest, calling MAP kinase ERK is now becoming more common than calling it MAP kinase. So this is just in case you do see those names used. Right, okay, so now where have we got to? We've got to the point where the activation of our receptor tyrosine kinase has resulted in the activation of our MAP kinase ERK enzyme. Okay, and we now want to see what is our MAP kinase ERK enzyme actually going to do. Well, basically, the activated MAP kinase enzyme will go from the cytoplasm into the nucleus. Okay, so it's going to go into the nucleus and it's going to phosphorylate molecules within the nucleus, which are transcription factors, and the phosphorylation is going to activate them. Okay, so it's going to go into the nucleus, and the first transcription factor that I would like to talk about is one known as ELK1. Okay, so here is ELK1, and I will colour ELK1 in blue here. Okay, so basically the MAP kinase ERK enzyme is going to come into the nucleus, and it's now going to phosphorylate ELK1. So I'll show this, as always, with a little blob on the side. Okay, so here is ELK1 now phosphorylated. Now, once you've phosphorylated ELK1, it's now going to act as a transcription factor. Okay, so let me draw my DNA here. So this double strand here will now no longer, well, these 2 parallel lines that I've drawn here will now no longer represent a phospholipid bilayer, but instead it's representing the 2 strands of DNA. I don't like drawing it intertwined. It always looks messy when I do that, so I'll draw them nice and straight. Okay, right. So let's say that this region of the double-stranded DNA here is a gene. So I want to explain to you what the concept of a transcription factor is. Okay, so let's say that this is a gene here. Okay, all eukaryotic genes have upstream of them a region known as the promoter region. Okay, so upstream of every eukaryotic gene, you have a special region called the promoter region, and I'll colour this in purple here. So this portion of the DNA box here is the promoter region for this gene, and every single gene will have a promoter region upstream of it. Okay, now the promoter region is not itself going to actually be translated. Okay, so it's not actually going to be turned into a sequence of amino acids, basically. However, it's extremely important in regulating how much of this downstream gene product, the product of this downstream gene, how much of the product of this downstream gene we're actually going to create. Okay, now the reason it's so important in doing this is that it is the place where the RNA polymerase II enzyme binds to. Okay, so RNA polymerase II is often abbreviated to RNAP2. Okay, now this enzyme is going to come and bind to the promoter region, and once it's bound to the promoter region, it will then work its way along the DNA, synthesizing a piece of mRNA. That mRNA will then go for translation and be turned into a protein. Okay, so if the promoter region has an extremely high affinity for binding to RNA polymerase II, i.e. it binds really well to the RNA polymerase II, then the RNA polymerase II will be binding there all the time. You'll be getting loads of mRNA being produced and therefore loads of protein. Okay, whereas if the promoter region has a very low affinity for binding to RNA polymerase II, then RNA polymerase II will hardly ever bind there. You'll get hardly any mRNA being produced and therefore hardly any protein being produced. So the promoter region can control the expression level of the downstream gene by determining how often RNA polymerase II will bind to it. Okay, right, and this is true for every single gene, basically. Now, a transcription factor, then, is a molecule which will bind to the promoter regions of a whole bunch of genes. So it will have a whole plethora of genes for which it binds to their promoter regions. Okay, generally it stacks up into the hundreds. Okay, so one transcription factor will bind to the promoter regions of generally around 100 genes. Okay, now what will then happen is by binding to the promoter region of these genes, it will change the affinity of that promoter region for binding to the RNA polymerase II. Now, some of the promoter regions it binds to within that 100 that I told you about, it will increase the affinity of RNA polymerase II for binding there. Okay, and others it will decrease the affinity of that promoter region for RNA polymerase II. Now, if the transcription factor binds to a promoter region and increases its affinity for RNA polymerase II, that means RNA polymerase II will bind to that promoter region more often. It will then work its way along the gene more often, and therefore will produce more pieces of mRNA for that gene, and therefore that mRNA will be translated into more protein. So, if the transcription factor binds and increases the affinity for RNA polymerase II binding there, you will enhance the expression of the downstream gene. If, meanwhile, the transcription factor binds and decreases the affinity of RNA polymerase II for binding to that promoter region, then you'll get RNA polymerase II binding less often, you'll get less pieces of mRNA being produced, and therefore less protein. Okay, right. So, every transcription factor will have some genes for which it enhances their expression, i.e. it increases the affinity of the promoter region for RNA polymerase II, and some genes for which it decreases their affinity for RNA polymerase II. So within the 100 promoter regions it binds to, at some it will increase their affinity, at others it will decrease their affinity. So every transcription factor will increase the expression of certain genes and decrease the expression of other genes. Okay, right. So this is what ELK1 is going to do. It's going to bind to promoter regions and change gene expression. Now, one of the genes then that it increases the affinity of, increases the expression of by increasing the affinity of that gene's promoter region for the RNA polymerase II is a protein called CFOS. Okay, now CFOS is actually itself another transcription factor. But in order to be an active transcription factor, it firstly has to dimerize with another protein to form something that can then bind to promoter regions of genes. Okay, and the protein that it binds to is a protein known as C-jun. So it forms a C-phos-C-jun heterodimer, like so. Okay, so I'll colour C-jun in red here. Okay, now, C-phos-C-jun heterodimers, like so, are going to act as transcription factors themselves. So they're going to bind to a whole plethora of promoter regions and change gene expression. Now, I'd also like to come back to MAP kinase ERK, because ELK1 is not the only target in the nucleus. It also phosphorylates a transcription factor known as c-MYC, okay, which is extremely powerful. c-MYC is a very, very famous proto-oncogene. It's very, very important in the study of cancer. Okay, and it's upregulated, or its function has increased in many, many tumours. It's often called the cell's most powerful mitogen. Its ability to make the cell divide is very powerful. Okay, so why am I coming back to c-MYC now? Well, c-MYC is also phosphorylated by MAP kinase ERK when it comes into the nucleus, and this will activate c-MYC. And the function of c-MYC is pretty much the same as the function of C-phos-C-jun heterodimers, which is why I've only mentioned c-MYC now. So this, if you like, is a more indirect pathway of doing what we're doing through this more direct pathway here. So I told you about ELK1 first, because ELK1 is going to increase the expression of C-phos so that we can form C-phos-C-jun heterodimers. Okay, but at the same time, MAP kinase ERK is also going to phosphorylate c-MYC, and now I can converge these two pathways together. Okay, because C-phos-C-jun heterodimers and c-MYC do pretty much the same thing. They change gene expression and they promote cellular division. Okay. In addition, they also are involved in differentiation as well. So they can also cause cells to differentiate into a certain type of cell. Okay, and this is done by changing gene expression. So remember, all the cells of your body have the same genes. Okay, so why is a neuron so different from a skin cell? It's because the epigenetics is different. The genes that are actually being expressed are different. Okay, so by changing gene expression, you can change the sort of cell you have. Okay, and it also causes proliferation, so you need large scale changes in gene expression in order for a cell to divide into two. Okay, and C-phos-C-jun heterodimers and c-MYC transcription factors, the changes in gene expression they produce is very similar, basically. Okay, right, so that's one of the pathways downstream of receptor tyrosine kinases. Okay, in the next video we'll turn our attention to a different pathway, which is the PI3 kinase AKT-mTOR pathway, which is also going to result in the activation of a transcription factor complex.